Hello, welcome to Module 4 of 4th week of our course on Introduction to Dynamical Models in Biology. In this module, we will try to make a mathematical model using ordinary differential equation for a small genetic circuit. Let us first look into the genetic circuit that we will model today. Here, this circuit is made up of two genes. Suppose the first gene is gene X and the other gene is gene Y. Gene X gives rise to protein X shown in the circle, and this X itself is a transcription factor, and that transcription factor X controls expression of protein Y from gene Y, and the control is negative. As you can see, we have a hammerhead. So essentially, X inhibits expression of Y. Y itself is a transcription factor which inhibits expression of X. It goes and binds to the promoter of X and inhibits expression of X. So what we have? We have two transcription factors, X and Y. Each of them have their independent promoters and each of them inhibit each other. So if I simply write it down, essentially if I have X and Y, Y inhibit X, X inhibits Y. In the diagram shown here, if you notice, we have shown a reporter gene fused with repressor 1. Repressor 1 is nothing but gene X. Now, many a time, to do an experiment based on genetic network, we require reporter gene like GFP, which will give us an estimate of level of gene expression. So the small network that I have drawn here has been actually tried in E. coli, and those people who created this synthetic network in E. coli, they had a reporter fused with protein X or gene X, so that X is expressed as a GFP tag protein, so that they can track how much X is produced by imaging or flow cytometry. Even if we forget about this GFP, we can still analyze and understand the behavior of this system. In essence, this system is made up of mutual repression. X is repressing Y, Y is repressing expression of X. So in other words, as it is a mutual repression, so it is a repression of repressor. Y is a repressor of X and X is repressing Y. So it is equivalent to have a positive feedback. So now we want to understand the dynamic behavior of this system, and we will create a ODE based model. And if you remember, we have to create the simplest ODE based model. One assumption that I will make here to simplify our model is that the transcription rate is very fast, and that's why the mRNA reaches steady state much before the protein can reach steady state. So the assumption that I am making is I am considering faster transcription, and that leads to steady state for mRNA. Once I have made this assumption, that means I have not to write a ODE for mRNA for X and mRNA of Y, because mRNA of X and mRNA of Y has already reached the steady state at very early time point. So I will have only two ordinary differential equations, one for protein X, and I have to have another ODE for Y. So if you remember, when we discussed in a particular module how to create ODE for a transcriptional circuit, we have discussed this aspect that many a time we can consider transcription to be very fast, and we can simply remove the ODE for that. And we can combine both transcription and translation together to make a clubbed equation. That's what I am doing here. So dx dt, the first ODE that I have, that is the rate of change of protein X concentration, is equal to K1 into 1 by 1 plus Y divided by H1 to the power n. So what I have considered here, I have considered a negative Hill function, because Y is inhibiting X, and usually this type of inhibitions are non-linear and follow sigmoid behavior. And if you remember, a negative or inverse Hill function is a very good option to model this type of system. So what I have used here, I have used a inverse Hill function to represent the inhibitory effect of Y. Next term in the ODE is minus Kd1 into X. Kd1 is the rate constant for degradation of X. So what I have in general for dx dt, the first term is production K1 into the negative Hill function minus Kd1 into X. The same thing is for Y. I have dy dt, rate of change in concentration of the protein Y. Remember, I am not writing down a ODE for mRNA of Y separately. I am simply writing down the ODE for the protein Y. So, rate of change of concentration of Y protein is equal to K2, the maximum rate constant for production, into a Hill function, which is again a negative Hill function with X as a controller. So, what I have 1 by 1 plus X divided by H2 to the power N2. So, that will control the rate of production of K1 in a sigmoidal fashion or inverse sigmoidal fashion minus Kd2 into Y. So, if I just brief what I have used inverse Hill function for both the ODE so that I can have negative control over the production of X and Y in a nonlinear sigmoidal fashion, and the second term in each ODE is nothing but first order degradation. Here H1 and H2 are Hill constant and N1 and N2 are Hill coefficient. So, this is my model, and now I want to analyze this model to understand the behavior of the system. Before I jump into it, let us simplify the model a bit further so that it will be easy for us to analyze here in this class. See, I want to understand the effect of rate of production K1 and K2, there is a maximum rate of production of these two proteins, and the effect of Hill coefficient like N1 and N2. So, what I have considered to simplify this problem is that I have considered H1 and H2 both equal to 1 and Kd1 and Kd2 both equal to 1. Essentially, you can normalize all your equation with respect to the rate of degradation and the Hill constant. That will simplify our problem. So, if I consider the H, the Hill constant equal to 1 and Kd1 and Kd2 equal to 1, I get the equations given here. You may have considered some other value of H1, H2 and Kd1, Kd2 that will not affect the way we analyze the system. The techniques that we will use remain same, only thing you have to do bit more numerical calculations, nothing more than that. So, I have removed H1, H2 and Kd1, Kd2 considering them 1, and I have these two simplified ODE 1 and 2. Now, I want to analyze this system of ODE. If you remember, we can analyze the system of ODE many ways. The first thing I will do here, I will try to understand the behavior of this system using phase plane analysis. To start a phase plane analysis, you should have a phase plane drawn. So, here I have two variables x and y, and then I will plot the null lines. So, let us first analyze the null lines of this system. So, first get the x null line. How do I get x null line? The ODE for x is dx dt equal to k1 into 1 by 1 plus y to the power n1 minus x. This is the ODE for x. So, I will put dx dt equal to 0 to get the null line. Once dx dt is equal to 0, then I can arrange that k1 into 1 plus y to the power n1 minus x is equal to 0. From this I am getting this one. So, once I have this, then I can separate out x and y on the both sides of the equal to sign. That is what I have done here. So, I have x equal to k1 into 1 by 1 plus y to the power n1. Obviously, this is a sigmoidal function. You can easily understand. This is a negative sigmoid function. So, now I will look into y null lines. For getting y null clines, what I have to do? I have to take the ODE for y. So that's what I have taken here, the second ODE. Then I have to make dy dt equal to 0. That's what I have done. If I have dy dt equal to 0, then these terms will be equal to 0. So k2 into 1 by 1 plus x to the power n2 minus y is equal to 0. So again I will separate out x and y on both sides of the equal to sign. So I get y equal to k2 into 1 by 1 plus x to the power n2. Again this one is a sigmoid function. So now I have two equations, x equal to something, y equal to something. These are x null cline and y null cline. So I will draw the phase plane plot. Now remember, in these two equations, k1, n1, k2 and n2 are parameters. That means to draw this plot, I should have numerical value for k1, k2 and n1 and n2. Let us try this. What I have drawn here in this screen, x in the horizontal axis, y is in the vertical axis. So this is my phase plane. In this plane, I will draw the null cline for x and y. As I have said, I have to take a numerical value for k1, k2 and n1 and n2. I have taken those values. k1, k2 I have taken equal to 3. n1, n2 also I have taken as 3. My null clines are given here. My x null cline is this one. My y null cline is this one. Let us try with y null cline. Y null cline is y equal to k2 into 1 by 1 plus x to the power n2. So obviously it will be a negative sigmoid function having a behavior like this, x versus y. If I plot, it should fall like this. So it will be a negative sigmoid or inverse sigmoid. And here it will intersect the y-axis. So when x equal to 0, y is equal to k2. So here k2 is 3. So I can see here this yellow line is my y null cline, and that is intersecting the vertical axis at 3. So that point x equal to 0 and value of y is 3. And then this null cline follow a sigmoidal fall with the asymptote here at the end. For x null cline, the plot will be just opposite. When y equal to 0, x equal to k1. And again k1 is equal to 3. So that means this pink line I have drawn here is the x null cline, and that intersects the horizontal axis at k1 equal to 3. And then it goes sigmoidally, and here I have the asymptotes. So I have two sigmoidal things. Both of them are inverse, but they are in the opposite direction. And as you can see, they are intersecting at 3 position as I have marked by red dot. If you remember, intersection of two null cline, that is intersection of x and y null cline, are nothing but the steady states. So that means when k1, k2 equal to 3, n1, n2 equal to 3, I have 3 steady states, because the null clines have intersected at 3 positions shown by these red dots. Now let us change the value of k1, k2 and n1, n2 to see what happens to these steady states. What I have done here, I have kept k1, k2 equal to 3, but I have changed n1, n2 to 1. If you remember, in the Hill function, n1 is the Hill coefficient or n2 is the Hill coefficient. If I have Hill coefficient bigger than 1, then only I get sigmoidal behavior. Otherwise I get a hyperbolic behavior. So here n1, n2, the Hill coefficients are not bigger than 1. That means I will not get a sigmoidal behavior, rather I will get a hyperbolic behavior. And when x equal to 0, y will be equal to k2. That means this is my y null cline, the yellow one. So when x equal to 0, it is intersecting at y equal to 3. And as you can see, this is not a sigmoid, this is a y nullcline is a hyperbolic one. Similarly, x nullcline is this pink one, and it is also hyperbolic, and it is intersecting the horizontal axis at 3, because when y is equal to 0, x is equal to k1. So k1 is 3, so it will intersect at this position. So I have two hyperbolic curves, and as you can see, they are intersecting only at one position. So that is my steady state for this system. That means when n1 and n2 is equal to 1, this system has only one steady state. Let us change the values further. Now I have kept n1, n2 equal to 3. That means I will get sigmoidal curve for nullclines. But we have changed k1 and k2 equal to 1. That means the rate of production has been reduced. So when you change the rate of production of the system, what will happen? The intersection point with horizontal and vertical axis will change. So now they are 1, not 3 anymore. But y nullcline is still a sigmoidal one, negative sigmoid one, and x nullcline is also an inverse sigmoid one. And as you can see in the curve, they are intersecting at only at one point shown by this red dot. So that means this system has only one steady state. So if in the last slide I have shown, if I reduce the system's nonlinearity from a sigmoid to hyperbolic one, I get one steady state. If I reduce the rate of production of the proteins, then also I will get one steady state. When both the rate of production of proteins and the sigmoidness or nonlinearity of the system or the promoters matches, I get 3 intersection or 3 steady states. Let us consider a situation where one promoter is more active or one protein is produced more than other. So in that case, k1 and k2 should not be equal. So I consider k1 equal to 1, k2 equal to 3. That means y is produced more than x. And I have considered the nonlinearity of the promoter function as sigmoidal one with n1 and n2 equal to 3. So if I draw the nullclines, this is my y nullcline, the yellow one. It is a sigmoidal one with a intersection of the vertical axis at 3, because k2 is equal to 3. For the x nullcline, the pink line, it is sigmoidal one, but it will intersect at 1 with the horizontal axis. Now as you can see in this plot, there is only one point of intersection between x and y nullcline, and that is shown by this red dot. So that means when I have an unbalanced system, both the promoters are following a sigmoidal inhibition, nonlinear inhibition, but one protein is produced more because maybe of the promoter activity or ribosomal activity, ribosomal binding site activity. So one protein y is produced more than the other one at a higher rate than the other one. Then again I don't get 3 steady states, but rather I get only one steady state. Now till now I have drawn only the nullclines. Obviously in phase plane analysis I have to draw the trajectories and the phase portrait, and that's what I have done already here. So this one is for the phase portrait for the system when your k1, k2 are 3, both are equally producing at the higher rate, and n1 and n2 are 3, that means both of them has sigmoidal control. I have 3 intersection points shown by these red dots. If you follow this arrow, you can easily see if I start somewhere here, I will follow this arrow and eventually collapse on this steady state. So all arrows near this steady state, near 3, is actually collapsing at that point. So that's why it is a stable sink node. Same behavior happen where I have another steady state near x equal to 3, as shown here by this red dot. If you start from anywhere, suppose here, then you will follow these and then you collapse here. If you start somewhere here, you follow these and collapse here on the steady state. So again, this steady state is a stable one and sink node. So I have 3 steady state, 2 of them is stable sink node. What about the third one? This is my third node steady state, and as you can see, this has a saddle structure because the trajectories here goes towards it and then move away. Trajectories near it goes towards it and then move away. If you start from here, you will go towards it and move away. So that means this one is a saddle point. This one is a saddle point. So I have 2 sink node and 1 saddle point which is unstable. Now if you have this type of system, then what will be the behavior of a population of cell? Let us analyze that. So if I have a population of cell with some cell having x and y at t equal to 0, this is t equal to 0, this is my position of some cell. So as time will progress, these cells will move towards this steady state. I may have another few cells at t equal to 0, they were here, they will also with time will collapse here. You may have some cells at t equal to 0 which initial condition x and y values were like this, at t equal to 0, they will follow this path and they will collapse. Whereas if you have some cell at t equal to 0 here, they will follow this path and collapse at the other steady state. So you can see, I can simply draw something, a line like this. Cells in this region will all collapse to this steady state. Let us say steady state 1. Whereas all cells which are present here in this region at t equal to 0 will collapse at the second steady state here. So that means initially suppose I have a unimodal distribution of protein x and y, then if I allow the system to progress, eventually the population of cell will diverge in 2 populations. One population will go to the stable steady state of sink node 1, the other population will go to the stable steady state 2. So I will get a bimodal distribution. That means this simple genetic circuit made up of x and y who are repressing each other can give rise to bimodal expression of x and y starting from a unimodal system, given that the value of k1, k2 and n1, n2 are matching properly. Let's see if I change k1, k2, what I have done. Now I have reduced the rate of production for both the protein. Now you can easily see these are the null lines, pink and yellow line that we have drawn. The phase portrait is drawn here with all the arrows. The intersection point, the steady state is the red one. You can see all arrows are actually collapsing at this steady state. So this steady state is a sink node. So in this case, if the rate of production of K1 and K2 is equal to 1, which is less than the previous case where they were all 3, then I get only one steady state, and that is a sink node or a stable one. That means if I start with a unimodal system, I still get a unimodal population of cells. What if there is an imbalance between production? So we have considered K1 equal to 1 and K2 equal to 3 earlier. The sigmoidal nature is still retained. So I have these yellow and pink line as null line that we have drawn earlier, and I have only one intersection point here near x, y equal to 3 and x equal to 0, near that. Now this is the only steady state, and if you follow the arrows present in this phase plane plot, you can easily see wherever you start, you will actually end up at this steady state. Wherever you start at t equal to 0, you will end up at that steady state. So that means it is a stable sink node. Again, this phase portrait is showing that if I have a unimodal population where different cell has different expression, but I have a unimodal behavior at t equal to 0, then with time all cell will move towards a particular steady state, and again I will get a unimodal distribution. I will not have bimodal distribution. So these phase portrait analysis is telling me one critical point, that if I have x and y repressing each other, so it is a repression of repression which is equivalent to positive feedback, then if the rate of production of both the protein and the N1 and N2 Hill coefficient are correctly taken, then the system can have bistability, and that can give rise to bimodal population behavior. Whereas if in the same system, if I reduce the rate of production of the proteins, or if I create an imbalance in production of these two proteins, I will not get the bimodality. I will have a monostable system and my population behavior will be unimodal. Till now what I have discussed is essentially phase plane analysis, which is done graphically. Suppose I want to analyze it using JSIM, and I will recommend you to use JSIM to simulate the behavior of this system. I have already shown the code here. It is standard JSIM code where I have only two dependent variables x and y, and these are the parameters, and you can play around with this parameter. I have considered all of them equal to 3, k1, k2 equal to 3, n1, n2 equal to 3, and I have taken at t equal to 0, I have x equal to 1 and y equal to 0. So I have considered the cells that I am taking have more x than y at t equal to 0, and these are the ODE as we have discussed earlier. Now if I do the simulation of this with k1, k2 equal to 3, n1, n2 equal to 3, and as I said the initial condition is x equal to 1, y equal to 0, then if I plot the data after my JSIM simulation, I get this curve. So I have time in this and concentration of the proteins here. So x is starting from this 1, this black line is showing the behavior of x, and it is increasing and then reaching the steady state near 3. Whereas y is starting from 0, it is rising slightly and then again coming back to another steady state. So the steady state of x is higher than steady state of y. If I draw the phase plane from the same data, in this plot in the first plot I have time versus x and y, and the second plot what I am doing, I have plotting x in the horizontal axis, y in the vertical axis. So the same data is plotted here, the same data is plotted here, so I will got a phase plot. So if you see the phase plot, at t equal to 0, I have this position where x equal to 1 and y is 0. Then with time, the system moves where ultimately x equal to 3 and y is slightly higher than 0. So this is my final steady state. So this is my phase plot. Look at the phase plot that we have drawn just now in the previous slide, which has a phase portrait and a graphical method has been used. So you are starting from here. So if you follow the arrow, you will land up in this position. So I get same behavior as I have got by numerical simulation. Let us do the same numerical simulation, same code, but I change the initial condition. Now y is more. At t equal to 0, y is 1, x equal to 0. So if I do the simulation in JSIM, what I get, y, the red line, start at 0, at t equal to 0 at 1 it start, and then it rises, and then it saturates at a steady state at higher value, that is 3. X now starts at 0, rises slightly, and then fall back and reaches the steady state, which is close to 0. Again I can plot the same data in x and y axis. That will be my phase plot. So at t equal to 0, x is equal to 0 and y is equal to 1. As time progresses, y increases, x slightly increases, and then ultimately we reach a steady state where I have x is close to 0 and y is equal to 3. So this is final steady state that we get. If I look at the phase plot that we have drawn just before, a few slides before, so this is my steady state here, this is another stable steady state here. Now I am starting from this position, y equal to 1, x equal to 0, that is my t equal to 0 position. So if I follow the arrows in the trajectories, so I get this trajectory and I reaches this higher steady state where, this is a steady state where y equal to 3 and x is close to 0, and that's what I have got here also. So that means by numerical simulation, although I don't get the complete phase plot, but I get the same behavior that is expected by the graphical method. So let us jot down what we have learned in this module. In this module, we have shown a 2 gene based mutual repressor system. So I have a mutual inhibition circuit which has a nonlinear promoter control, and this type of circuit is sometimes called toggle switches. And we have shown that if the nonlinearity of the promoter control and the rate of production of the protein matches, then this system can have obviously bistability. If you change the value of the rate of production of the protein and the nonlinearity in the promoter, you changing the value of the Hill coefficient, then from bistable system I can have monostable system. That means the system has bifurcation. And this choice of monostability and bistability will depend upon maximum rate of production and the Hill coefficient. Hill coefficient is a measure of nonlinearity in the system. And as a bistable system can give rise to bimodal population behavior, so we can expect that such a system can give rise to population heterogeneity. And what I have shown in this module, that I can analyze this system using both graphical method by phase plane analysis as well as by simple numerical simulation using JSIM. That's all for this module. Thank you for watching.